Good afternoon. Please take your seats, and we are ready to start. So welcome. Thank you for joining us today for this event, Space Travel with Earth Wisdom. Looking very much forward to this. Before we get the program going, I would like to take a few of your minutes from the program to introduce the Hariri Institute, that is the host for this program. I'm Yannis Paschalidis. I'm the director of the Hariri Institute and the faculty in Engineering and Computing and Data Sciences. So the Hariri Institute is the home of about 10 centers and initiatives that you see on this slide. One of our signature programs is what we call the Focused Research Program. So this is an annual program. We have an annual call. We fund about 4 or so of these focused research programs every year. And the objective is to bring together teams that are highly interdisciplinary and facilitate research in the interface of computing and data sciences, which is the core of the Hariri Institute, with many other disciplines, and get these teams going in terms of having broader impact, but also enabling them to be more competitive in bringing larger federal or other types of awards at Boston University. So if you are interested, if you are at BU and you are interested in this program, the spring is the time of the year when we accept proposals. Feel free to go to our website. There are details there about the program and the requirements. So just to give you a taste of what we funded during the current fiscal year. So these are 4 programs that we have funded, and one of them is the first trip to Mars that Marianna, Felici, and Paul Withers are chairing, are the PIs for this program. So I will introduce Marianna and Paul as the PIs. So Marianna is a research scientist at the Center for Space Physics at BU. During her PhD, she worked on charged particles trapped inside the magnetosphere of Saturn, but since then has expanded her work to also include the Mars ionosphere. That is one of the goals, to study that, the goals of this FRP. And she has been involved, she's part of the Mars Atmosphere and Volatile Evolution mission, so-called MAVEN mission, and she's also working on the radio occultation science experiment called the ROSE experiment that studies how the Martian ionosphere changes with local time and season and how it responds to different events like dust storms and precipitating particles. So she received her bachelor's in physics from the La Sapienza University in Rome and a master's from the same institution in nuclear and particle physics, and a PhD from the University College London in space and planetary physics. Paul Withers, who is the other PI for this FRP, is a professor of astronomy, and he currently serves as the chair of astronomy at BU. His research interests include ionospheres and planets. These are extremely important problems, as we don't really understand very well what is happening, and the high-level picture, the chemistry, the energetics, the dynamics, and the electrodynamics. And I don't think we still can describe the interactions with the solar wind above and the lower atmosphere below. So Paul makes progress towards this overarching scientific goal by leading a series of experiments on spacecraft missions. He received his bachelor's in physics and MS also in physics from Cambridge University in the UK, and a PhD in planetary science from the University of Arizona. So please join me in welcoming Marianna Felici, that will give you an idea of what is the plan for today. Thank you.
Thank you, Yannis, for the introduction and for supporting us in this past year. So our hope is that today you walk away fantasizing and mentally building castles on Mars, but also more deeply in love with our planet. It's got forests that talk through fungi roots with tales that most of us have forgotten. So with the promise of a sci-fi future with a space travel supported by AI, now more than ever it is important to remember where we come from and to connect with the life that there is on Earth. So today's event is organized by Boston University, the Hariri Institute for Computing, and is supported by the Departments of Astronomy, Earth and Environment, Computer Science, and Electrical and Computer Engineering. We thank them for making today possible. So this is what you signed up for. So in the first part of today, we will tell you what our team has been up to, from sky to the surface. And then in the second and third part, invited speakers from sky to the ground and panelists from ground to the sky will give us some food for thought. In between and after, some actual food, which you're welcome to take with you. So if there is anything you like and you see we have excess of, we have takeout containers and bags that you can use to help us consume everything. There are markers over there. So this institute was built with some walls on which we can write. So that's fun. So if you make new friends today, there are markers there to collaborate with them. And please fill the back of your name tags, because we're going to use that later. So there are pens at the registration desks to do that. So these are some housekeeping announcements. Today's talks are being recorded and will be available next week on the Hariri Institute YouTube channel. We ask those who have questions to hold them until after each presentation. For those in person, there is a microphone over there, and you can either walk up there or we can take it to you. And for those online, you can enter your questions through the Q&A feature on Zoom. We have Zoom moderators, and we'll try to keep up with the questions and the talks as well. We have a timekeeper, and I suspect a trapdoor. So we need to kind of keep time and move this along. So we might not get to all the questions, but if you email us, we'll try to do our best to follow up with you. So we have a brilliant line of researchers today presenting. Thank you so much for coming, everyone. And you will hear from all of them shortly. So about this project, we started from we wanted to holistically support human missions to Mars, which is a private sector goal for 2030s, but also NASA's goal to land humans on Mars in the 2030s. So we started from modeling the thermosphere and the ionosphere using data, mostly from MAVEN. We're going to clarify all of this later on, so don't worry about the terminology there. That we started thinking about communication and navigation system while doing new science and also monitoring the solar wind activity. And we started thinking about what microbes could serve as a source of food and nutrients, but at the same time can be applied in the fight against the global warming. This wouldn't have been possible without the Hariri Institute, so this seed funding was actually pivotal because federal grants do not cover all the components to be grown simultaneously from the ground up. And they made possible that 6 faculty, 5 students plus the software application and innovation lab at the Hariri, and you're gonna hear from them as well, plus 1 research scientist, come together in 120 plus hours of meetings this year, so it was a lot. And you'll see, it was a lot of collaborating, and you'll see what we've been up to in a moment. So this is only the core science and engineering team from now. And, oh, actually, I should have done that. So if you see yourself up here and you're in the room, can you please wave so that everyone can chase you and track you down? There you see Prakash there. Awesome. Thank you. So yes, so chase us during the breaks. So team speakers today, myself again, just a few minutes, I promise. Then we have Brackney, Josh, Dylan, Ilya, Zoe, and Will. And for Will, please follow him on the info booth over there because they're gonna show you the gigantic screen at the back of the room, but they're gonna show you what the Hariri software engineers have been making for us. Yes, up in the air, that would be me. So this is the basics. The sun is over there, the entire solar system that way. Distances aren't in scale at all. The Earth is there, and Mars is the next rock from the sun, and it might be just a close hop. It isn't, and you're gonna hear why. So this is a close-up of Mars. There are signs of past liquid water that left the planet somehow, and this is like answering and understanding why the liquid water left the planet was the primary goal of the MAVEN mission that was designed primarily to study the atmosphere and atmospheric escape. You're gonna hear about MAVEN data a lot during these talks, the various talks, so I thought of mentioning it right in the beginning. Other words you're gonna hear are ionosphere, thermosphere, neutral atmosphere. So here on this is the left, yes. There is a schematic. So for atmospheres, at the bottom you have a lot of neutral atoms and molecules. Right at top you have a lot of solar photons that get less and less as you go down because they get absorbed. When these two curves, like the trend of photons with altitude and the trends of density and molecules, density of atoms and molecules with altitude cross, that's when you have maximum ionization, which means one electron is knocked out, not for every atom and molecule, but some of them lose their electrons. And so what you have left is positive ions. So you see on the plot on the right over there, CO2 plus, O2 plus, and a lot of electrons that are just by themselves that will give you this curve over here, and that's a vertical electron density profile. So the science that we've been doing with our trust was since we work on this theme, so the radio occultation science experiment ROSE, whose PI is Paul sitting over there, and I'm the deputy PI. This is the data we collect, and this is an instrument on MAVEN. So what we did was parameterizing and labeling hundreds of these profiles, and they aren't all so pretty, like these ones over there. And we applied machine learning algorithms to find new trends in the data or things that we couldn't intuitively separate for ourselves. And so we checked what the machine would see. And now we're utilizing neural networks, and for that, Prakash, sitting over there, and Brackney have been helping. So I'm just gonna stop talking because I've been here quite a bit now, and I'm gonna pass the ball to Brackney, who's gonna tell you everything about neural networks and neutral atmospheres.
Thank you. Hey, everybody. Today I'm going to be discussing a neural net model that I've been working on that helps predict the neutral atmosphere, but I'm going to go through some background first. So, the neutral atmosphere of Mars. This is a temperature-altitude profile of Mars and Earth. The first most glaring difference here is that the Martian atmosphere is colder. Not visible here is that it is also 30 times less dense and has a dramatically different composition. It's mostly carbon dioxide. As with all atmospheres, it has many layers, and these layers have really important properties that we as scientists need to study and engineers need to be able to account for. The lower atmosphere is generally controlled by dust storms and gravity waves, clouds, etc., all kinds of wave and cloud activity. The upper atmosphere is sensitive to solar radiation, as it is essentially the planet's direct interface with the space environment that is controlled by the sun. The ionosphere, the charged atmosphere, as Marianna discussed, gets all of its charged particles by ionizing the neutral atmosphere, and so whatever changes the neutral atmosphere changes the ionosphere. So what's the big deal here? Is it useful to us? Can we breathe it? Can we use it for parachutes like we do here on Earth? No. It's far too thin and it doesn't have the right composition. So, okay, so it's really thin. Is it thin enough that it's really simple and we can basically ignore it? Also, no. It has just enough density that we need to account for it pretty much all the time. It is highly variable and highly complex, and it affects all of our operations at Mars in a lot of different ways. So here we just have a simple example of a satellite and a hypothetical human presence on the planet. And I'm going to go through all the different ways that the Martian atmosphere can impact both of these systems. So first off, communications. The ionosphere can block communications, which is generally bad. It's much thinner than Earth's atmosphere, and so dangerous radiation from the sun can come and reach the surface, but how much reaches the surface varies due to the atmosphere. So having a good idea of how much radiation is going to reach the surface at any given time, very important. It is useful in slowing satellites in a process called aerobraking. Essentially, you dip the satellite's orbit deep into the atmosphere. This causes drag, slows the satellite down. If the atmosphere is highly variable, you need to be able to understand how that variation will affect this technique. And lastly, if you're observing in one spot, you don't know what's going on in another spot, say directly above your, brand new Mars colony. So you need a model to account for all of these things, and that's what I've basically built. So I'm using a different instrument aboard the MAVEN satellite called NGIMS. It is a mass spectrometer. Essentially, it takes in small samples of gas, determines the density and the composition of the sample of gas, and it does that continuously throughout the orbit. From this, I can get a pressure-altitude profile, and also when and where and in what conditions that profile was taken. Using a known relationship between pressure and temperature, I can derive the temperature altitude profile from the pressure profile. In addition, I can grab solar conditions from other satellites that are just continuously monitoring the sun. So using the profiles from NGIMS and the position and time that they were taken, along with solar conditions, I can produce a neural network model. If you're into neural net infrastructure, I have it listed down there. So this model can take in position and time data and export solar conditions. Position and time data and solar conditions and export the corresponding predicted temperature and pressure. So how well does it work? It does a pretty good job. So in this, I have the pressure on the left and temperature on the right. On the left, you can see that the pressure profile is pretty solidly well predicted, the blue versus the orange. The pink line on the other side is created by taking the predicted pressure profile, applying that same physical relationship to temperature, and deriving a physically connected temperature from the modeled pressure. And while the predicted temperatures aren't great, the physically connected predicted temperatures are a much better relationship with the real, with the real temperatures in blue. So what can we do with this? Well, first things first, I can feed it a 1 degree resolution latitude-longitude map of the entirety of the planet and ask it to predict the pressure at 200 kilometers everywhere for a particular time of day, time of year, particular solar conditions, etc. That creates this plot. So the x and y axes here are latitude and longitude, with the top x-axis corresponding to the time of day at that particular longitude. This model does a pretty solid job of capturing the general behavior of the pressure. So we see this low pressure at night due to it being relatively cool. We can see the warming throughout the day, throughout the morning, and then in the afternoon and throughout the day, we can see much higher pressures due to higher temperatures. The other thing we can do pertains to that predicting outside of observations. So there is another instrument aboard the MAVEN satellite that can also predict pressure and temperature. It is called IUVS. So NGIMS is what we call an in situ instrument. It collects data right next to the satellite. However, IUVS is a remote instrument. It looks somewhere, it collects light, and we can determine information about the place that it's looking at. So a natural question arises, since you have two instruments that are observing the same phenomena in different places, what would NGIMS see where IUVS is looking? Now, because I have a model that functionally behaves like a synthetic NGIMS, I can actually answer that question. So if I feed my model the IUVS position and time data along with the solar activity recorded at that time, I can get what the NGIMS output would look like for that position. And it actually shows really, really nice agreement. We can see that the predicted orange output and the IUVS profile line up quite nicely in their admittedly limited overlap. This is not only a great demonstration of this technique for potentially being used to cross-calibrate instruments, but also just a way of predicting what the conditions of the planet look like given a limited set of measurements. I'll take any questions because I think I have a couple of minutes, but thank you. Sorry?
I'm going to get the mic here.
Thank you.
How do you see this being applied for predictive use?
So a more advanced version of this model could be used to say, let's say the closer orbiting solar monitors detect a large CME coming towards Mars. We could then use the model, input okay, so we know the CME is going to be this strong when it reaches Mars. We know it's this time of day above our colony. It's this, these particular conditions, this particular location. What does the atmospheric response look like? And so we can then go from there and figure out, okay, how hazardous is this to our mission? Is it going to interrupt communications? Is it going to potentially risk our equipment or the people that we have there?
Hello, I want to know which kind of neural networks do you use? And also, what's the input of neural network and what you want to predict?
So it's just a simple, fully connected neural network. The node structure and the inputs are right here. So I trained it on these three inputs, so the positional data from the satellite, the pressure temperature profiles, and then also solar conditions. The model can take in that positional and solar data and output the pressure and temperature. The structure is listed right there. I have, I think, 5 hidden layers and a couple of dropout layers.
And the inputs are solar, wind proxies, and the output should be the proxies around Mars, right?
So the inputs are the position at Mars you want to predict, and the solar conditions. I'm actually just using sunspot number right now, but more complicated model, we'll use a more complicated measurement. And then the output is the pressure and temperature at Mars. So actually we're looking directly at the response as opposed to extrapolating solar conditions. But my time is up, so thank you all.
Thanks.
There you go.
Now we have Joshua Sumter.
Hey, everyone. All right, as Marianna stated, I'm Josh Semeter, and I'm currently serving a term as director of the BU Center for Space Physics, and I'm happy to try and contribute a few ideas around what kind of infrastructure we need to place in orbit around Mars to support what's coming. And what's coming is the exploration phase, the human exploration phase of that target, of that destination. The expansion of Western civilization across the Americas was preceded by an exploration phase. And that, of course, had a lot of thorny issues associated with it, the behavior of the explorers, the behavior of Western civilization. And although we're not expanding into a colonized environment at Mars, that you can rest assured we're going to have our own thorny kind of ethical and moral issues associated with whatever we do with Mars and how it relates to our societies here on Earth. But we're here to talk about the technical aspect of things. And so there is an analogy to the expansion, the westward expansion of into the Americas in that it's a place that we're going to where we have an unknown environment and we're going to kind of figure out what that environment is by going there blindly. We have, of course, the advantage, the twist, if you will, of having the ability to have sent robots to the Mars environment for many, many years now. So we actually know quite a bit about what's happening there. And each time we send a satellite there, typically up until recently, it's been a standalone mission, meaning it's everything that you need is on the satellite in order to know or control its navigation and to telemeter its data back through the deep space network. We've recently done things like with MAVEN, where we actually deploy a radio relay on there so we can communicate with surface elements and actually serve as a relay to get the data back to Earth. What makes most sense, of course, is to, in anticipation of a full-out exploration phase, we should deploy an infrastructure to Mars that does communication, navigation, and we're going to argue scientific research from the same set of platforms. Okay, so let's start with knowing where you are. Let's start with global positioning system. What does GPS look like on Earth? What does it look like on Mars? To kind of remind you of how things look here, this is what you have if you want to think about a rotating planet with a constellation of satellites in geostationary orbit around it. Then your view into the satellites that are above the horizon changes as you move. GPS was cleverly designed to make sure you can see 4 satellites, and those are indicated by the red lines as you rotate around the planet. Turns out 4 satellites is exactly what you need at minimum to know where you are. You need 3 to determine position, but there's also an unknown timing alignment that has to be corrected for. Okay? Now, I want you to keep this view in mind as we move forward, because I'm going to actually sort of begin with the argument that we should think about GPS and GNSS, especially at a place like Mars, in terms of its scientific potential, because we have information in the propagated radio waves, in addition to its operational necessity. Okay, so if we look at Earth versus Mars, I should mention up front that this is meant to be a relatively non-technical talk, and we have a paper forthcoming that kind of makes some recommendations that are more technically complete. At Earth, we have forest canopy, buildings, precipitation, water precipitation. In Mars, we have an open rocky planet, and so we don't really need to go up to the sort of gigahertz or frequencies that we use at Earth. In fact, we can do a lot in terms of positioning using UHF frequencies. And what that means is that they're going to interact with the ionized matter surrounding the planet in a different way, in a way that's actually more scientifically effective. So that's one notion, is that we want to think about perhaps UHF frequencies in our GPS solution at Mars. Accessibility of the space environment is high here at Earth. It's pretty easy to put things into low Earth orbit. We have no ground-based infrastructure at Mars. Constellation deployment is logistically challenging, favors combining research and operational objectives for efficiency and scientific efficacy. The per spacecraft deployment cost at Earth, just the deployment costs, it's low now, 100,000 will get you into space. and a ride share per spacecraft deployment at Mars is going to be around 100 million. So, this favors, can we get away with smaller constellations, or do we need 24 or 30 satellites like we have in GPS? This is the basic idea of the network that you would want to put, I believe, at Mars. A minimum of 6 microsatellites. That actually allows you to get a lock on your position with 2 hours, within a 2-hour window, considering the orbit of the satellites. And more quickly after that, after you isolate, we have really nice engineering algorithms. If you isolate a person's location, if that's your initial guess, you can run things like adaptive filters to figure out where they have moved to without another 2-hour window, from scratch window. The idea here is a microsatellite constellation that has UHF and also X-band, which is 10 gigahertz for communication directly back through the deep space network. The UHF being primarily used for the landers, rovers, and so forth, and for the atmospheric science, which we'll discuss. And then there's some small number of aerostationary, we say geostationary here at Earth, I guess aerostationary is correct for Mars, that's got a high rate, a high capacity, 100 gigabytes per day connectivity. Okay, so ionospheric research with GNSS. It's quite simple. Long ago they figured out that there is this ionospheric delay that causes an advance of the phase and a delay of the group velocity, and it's very knowable if you can just simply transmit at 2 closely spaced frequencies and fit a line between the two. You can remove the ionospheric effect and increase your GPS accuracy. The part that you remove is in fact proportional to what's called the total electron content. It's the line of sight integrated plasma density between satellite and receiver. It's a simple equation, it's a simple parameter to calculate. It's got a lot of really interesting nuances in terms of interpretation, but this comes for free when you deploy a constellation of navigation satellites. Okay? And just to give you an example, a really nice terrestrial example, imagine now that TEC observation is available from 1,500 receivers on the ground, all looking into the same satellite. That's exactly what we have in Japan, a highly seismically active region, and so they really want to know things about the geography, the geology shifting and so forth. And so they've deployed this immense network of these receivers. You can do careful analysis of this TEC parameter and actually make for yourself very highly detailed movies of events that don't originate in the ionosphere at all. So this is an event, this is the tragic Tohoku earthquake, propagates its waves from the epicenter up through the ionosphere, and the imprint on the ionosphere makes the whole thing visible. Neutral gases are hard to see. You need specialized mass spectroscopy and so forth to see them. Electrons and ions are electromagnetically reactive, and so you can easily see them and create pictures like this. So this is just a flavor of what we could do at Mars scientifically. This is a movie, so yeah, the neutral atmosphere is difficult to observe. The ionized state of matter is easier to observe due to its electromagnetic reactivity with a plasma. And the big question at Mars is atmospheric escape. And so the GNSS constellation that we deploy to Mars could actively contribute to providing spatially resolved measurements of the plasma environment. The plasma environment is really the key, okay? And so this is a test particle simulation of the solar wind interacting with the Mars environment, creating a bow shock that eventually what you're seeing here is simulation of the plasma, mostly O2 plus being extracted from the planet. There's a complex interplay between the plasmas and the neutrals occurring in the Mars atmosphere that creates this outflow of matter. Okay? So one of the keys is the ionosphere. I think the ionosphere is the reservoir, and we can image the ionosphere through a constellation of satellites that is also deployed for navigation and communication purposes. And so this is the last slide here. There's a lot more to discuss with this, and I'd be happy to do so throughout the course of this meeting. These are some technical considerations. We need uniform global coverage. We need high capacity, low latency communications. Should be focused on equatorial regions where we think we'll probably land and where the Mars sample return region will probably be. We need a robust system. We really got to think about that. We can't have the whole system fail because one of the 6 satellites failed. So there needs to be backup. There needs to be durability. We got to put propellant out there so that we can have at least a 5-year time span where it works. And then there are other scientific considerations. You know, do we want to design a system that could be used to investigate the moons of Mars, Phobos and Deimos? Yeah, I'm all done. Space weather monitoring and prediction is another scientific objective. Thank you. All right.
So now we're down to the surface, and that's welcome, Dylan Mankel.
All right, so I'm kicking it off. What do we actually do when we get to Mars? How do we actually keep astronauts alive? How do we let them build new structures and materials using the stuff we can find on Mars? And so hopefully, at the end of this talk, I can convince you that we can use microbes from extreme environment analogs to grow bioplastics on Mars. And so when I refer to an analog environment, what am I talking about? So analog environments are simply places here on Earth that resemble some aspect, some condition of an environment that we want to study that we can't otherwise reach. So we can use places like the Atacama Desert to study how microbes might survive or how life might survive in the dry, arid environments on Mars, or we can use deep sea hydrothermal vents to study what vents might look like on ocean worlds of like Europa or Enceladus. And so picking the right place to study here on Earth is just a matter of matching the conditions in these far-off places. And so, for instance, on Mars, liquid water may have been, or actually may be present on Mars. And so here's a picture, amazing picture of the Jezero crater. I mean, it doesn't look too far off from river deltas here on Earth, like the Nile. And so we've actually landed the Mars 2020 Perseverance rover in the Jezero crater to search for life, search for signs of life and past life. And from satellite imagery, these minerals called olivines have been present throughout this region. You can see the Jezero crater right there. And the amazing thing is when you mix these two together, olivines and water, they can react together through this process called serpentinization. It's this metamorphic process that may have been or may be occurring on Mars today. And the amazing thing about this process is that this reaction can provide a chemical source of energy for microorganisms. So you simply mix these two together, they react, they create a host of byproducts, but the one that we really care about is the hydrogen and the hydrocarbons that can be produced from this reaction. This process also creates hydroxide ions, so it creates these ultra-basic conditions that are very characteristic of serpentinizing systems here on Earth. And so where do you actually find this on Earth? So one of these places is called the Cedars. There's many places here on Earth, but the one that was first discovered, or at least first to have active serpentinization, is the Cedars in Northern California. Sorry, I should go back and say that olivines are really abundant in the mantle, but why would these materials be anywhere near the surface here on Earth? So at some point in Earth's history, in California's history at least, there would have been two converging plates, and as they converged, the oceanic lithosphere, which would have been rich in these olivine minerals, would have been scraped on top of one of the continental plates, leaving it exposed where water can seep down into the subsurface, react with that olivine-rich rock, and cause serpentinization, inducing this reaction. And as the serpentinized fluid makes its way back to the surface, through cracks and fissures in the rocks, it creates these really high alkaline springs that we can see here at the Cedars. And they're very easy to spot because, as I had mentioned, they're very ultra basic. So any CO2 that actually dissolves in the fluids basically immediately crystallizes as calcium carbonate, which encases many of the pools. And it's actually very clear that this process is still occurring today, even after 50 years since the cedar had been discovered. So these are those gas bubbles that I'm talking about that are being released from serpentinization, and you actually see the constant fluid flow coming from the subsurface. And despite the extreme conditions at the Cedars in these pools, we're talking about pH 11, pH 12, much higher than your common household bleach. There are many microbes that are home or make this place their home, and they can utilize that hydrogen released from serpentinization as an energy source. But the one that I most care about, and what I find most interesting because it's actually been isolated, is this genus called Serpentimonas. And so Serpentimonas actually stores carbon as polyhydroxyalkanoates, or PHAs. They're these polyester carbon-rich molecules. And the amazing thing about those is those polymers are actually promising alternatives for other industries for a wide range of products, including petroleum-based plastics, as well as biomedical and environmental applications. And so we have this microbe from a Mars-like environment that can do these awesome reactions to produce these PHA polymers, and it can grow autotrophically, so we don't need to ship a ton of sugars like we would for many other microbes that do this process. So it can grow on its own using the resources we find on Mars. So hopefully, or my hope is that we can ship this microbe with future astronauts so they can actually grow this microbe using the materials on Mars to create all these products in situ. And so to do that, we want to understand a little bit more about that PHA production, because this microbe is very new. It only has a handful of papers on it, but we really don't know much about how much PHA produces, so we really understand more about that mechanism. So to do that, we developed a preliminary genome scale model. So we took that model, we assembled it, we annotated it, so identified any known enzymes across that genome, and we built this draft model, which is part of it is what you see on the right. So anything in blue are enzymes that we have found in that genome. And then we also gap filled it using information we know about what it should be able to grow on. And the model is actually consistent with many of the published experimental results, but it has yet, unfortunately, to grow in our lab. But that is the hope over the next couple of months, is actually to get it to grow and actually test that PHA reduction across the number of substrates that have been tested from the literature. And as Ilya will eventually discuss, the goal is to then not only use the PHA production that we measure in the lab and compare it to what we can simulate on the computer, but then we can also feed in growth rates and the metabolome into this software called Comets. And that can actually simulate the microbe on your computer, simulate it over time, so using conditions that we wouldn't otherwise be able to produce either in the lab or, say, on Mars. We can test how well this microbe might grow or produce PHA wherever we want to in any conditions we might like. So Ilya will discuss that right after me. But the ultimate dream is that we can ship this microbe out with future astronauts so they can build structures and tools just using this humble microbe and the materials they find there. Thank you. Sure.
What do you mean we can just ship it? Just a vial of it?
Yeah.
And then
Yeah, so most microbes we store, at minus 80, in the freezer, essentially. And so you could revive it back on Mars using either materials you bring or materials you might find on Mars.
Mentioned serpentization, was it? Is still happening on Mars, maybe?
That's a great question. I'm not sure. I mean, we haven't exactly found like a ton of liquid water to promote a lot of serpentization, but we also haven't really drilled, deep into the subsurface. So the cedars, for example, all that reaction is happening deep underground. So it could be happening on Mars today, could be happening right as we speak, but I don't think there's any current evidence to show that it's actively happening. But the goal is that if we, say, bring a ton of water and we can mine all this rock, mix the two together and create serpentization just using the rocks there right now.
Cool, thanks.
Yeah.
You're testing with the cedars stuff? You haven't got Mars samples yet, right?
Correct.
Okay.
Correct. This is all the cedars.
So some of the sites are showing perchlorates in the soil, and I'm wondering what the implications of they are.
Perchlorates?
Yeah.
I'm not sure. Can you be more specific, or?
Well, I think they're adverse to microbial life, which is why I'm asking about that. But they're presumably a byproduct of photochemical reactions over the eons and are contaminating. I know they found them at the Viking sites, and I think they found them at some of the Mars exploration rover sites too. So I guess I'm really just asking the question of what those kind of contaminants in the soil might do to what you would attempt.
Like how they might affect the microbe?
Yeah.
I guess if they are inhibitory to the microbe, we could certainly clean up the rock that we might add as a media for the microbe. Yeah. Thank you.
Now we have
Hi everyone, I'm Ilya Dukovsky and I'm a researcher in Daniel Segre's lab, and today I'll tell you a bit about the modeling of microbial community activities that are happening in Daniel's lab. The focus of research in Daniel's lab are microbial communities in different systems, whether it's gut of humans or insects or the ocean, and what's more relevant for this project, the microbial community or the microbiomes as we call them in soil or even in the lab, synthetic communities that we create and grow in the labs. So microbial communities are very complex systems, and we would like to create models of those systems to get an insight into that complexity. Why are they complex systems? Well, first of all, there are lots of organisms, lots of species that are present in a typical microbial community, and those organisms are interacting with each other. They're interacting whether by competing for the food resources or by exchanging metabolites among each other. And we want to study them in their natural environment, so like this root system, for example, and that adds to that complexity. The spatial structure adds to the complexity of a community. And even in a Petri dish, here on the right, I'm showing you some colonies that were grown on a Petri dish, which are, fantastic. They're very complex. They are creating these really crazy shapes and patterns. So we want to capture some of that with our modeling. And so how do we do the modeling? Well, we start with the genome of the organisms. We sequence the DNA of organisms, and from that information, we can put together the list of all chemical reactions that are present in an organism. In other words, we create a model of the metabolic networks in the organism, and then we solve those networks. What I mean by using the flux balance analysis method, what I mean by solving is essentially this algorithm, the FBA algorithm, will give us the rates with which, or how fast an organism eats the food, how fast it grows, and then the organisms are also producing waste that they want to get rid of. They are secreting that. Now, what waste for one organism is a treasure for another one. That waste can be eaten by another organism. So there is a time dynamic of those interactions that we also want to model. We model that dynamics using the dynamic FBA algorithm, and here I'm showing you by solving the differential equations of the dynamics of the system. Here I'm showing you in green an organism that's feeding on the blue molecules there. Let's say it's a glucose. The glucose is being depleted. The green organism is growing and starting to secrete this red product, can be acetate, for example. And now that red product is feeding another organism shown here in yellow. So that's how we capture that dynamics over time with this algorithm. But we want to go a step further, and we want to capture the spatial structure. So we combine this dynamic FBA with the model of the biophysical model of the propagation of that biomass in space. And all that put together is packed together in our program, our software, COMETS, that stands for Computation of Microbial Ecosystems in Time and Space. So here are two examples of simulations that we did with COMETS. On the left, I'm showing you simulated growth of a culture in a well-mixed conditions. So this is essentially bacteria that's growing in a vial. At the first, you see that the bacterium is growing very fast. It's feeding on a preferred food, that's glucose. And then when it's running out of glucose, it starts to feed on another food. In this case, it's the monomer of chitin. Chitin is the stuff that insects are made of. So there is a shift from fast growth on a preferred food to a slower growth to a food that is not preferred. And the scientific name for that is dioxic shift. So in a way, this organism kind of eats the cake first and then switches to the salad. So, and you see that we capture that with our model. The blue curve is the COMET simulation. It fits very well the experiments that's shown in yellow. And I want to point out here that we did not inform the model that this is supposed to happen. This effect, this shift, is an emergent from the modeling, from the time dynamics. In other words, that information is kind of written in the genome, in the DNA of the organism, and the model shows that without us telling it explicitly that this is what's supposed to happen. A similar situation, somewhat similar situation we have on the right, when I actually simulate growth of a bacterial colony on a Petri dish. So this is a spatial propagation of the E. coli colony. The experiment is shown on below this curve of the diameter of the colony versus time, and the simulation is shown above the curve. And you see that we do also have a good agreement in the overall shape of the colony. And again, you see that the shape is not perfectly circular. It has these protrusions, kind of branching of at the growth front. And we reproduce that phenomenon in our model. And again, that's something that we did not explicitly tell COMETS that this is what is supposed to happen. This is also emergent phenomenon of the combination of the metabolic modeling and biophysical model of the propagation of the colony. All right, so COMETS is an open source written in Java, and it's free for academic use. Anyone can download it from our website and use it. It's written in Java, but we also created a Python interface and a MATLAB interface, so anyone who knows Python can easily use it, install it and use it. But we want to go one step further. As part of this project, in collaboration with SAIL, we are building a graphical user interface to make it a virtual lab. So this would be a user interface where somebody who doesn't know any kind of programming can easily go and perform experiments on a website by choosing options and for the models, for the organisms that they want to grow, choosing what media they want to grow them, or what kind of layout, was it going to be a petri dish or whatnot. To make this software available to a much broader audience of researchers who don't necessarily are familiar with any kind of computing. So this graphical user interface is done by the SAIL team, as I said, under leadership of Marianna and Will. And I would like to point out Hazim, Karina, and Colin, who actually did heavy lifting, did the software development. Thank you very much. And don't forget, we have a booth over there at the back where we can demonstrate the capabilities of Comets, and we can show you how it works. So feel free to stop by over there. I'll be there after this session. Thank you very much, and I think I have time for questions.
You show the model. Would it be going down when it runs out of food? Like it goes up and then it's kind of using non-food, but would it run out of food at some point and go down?
Yes, it will, and we do have a death in the model. In this case, I didn't include it because I didn't need to, but in principle, yes. Once it runs out of food, it will start to die, and we actually have that capability to simulate that in Comets.
Thank you. So my name is Zoe. I'm a PhD student in the biology department here at BU. And I'm going to be relying a little bit on the background that Dylan and Ilya have given on genome scale metabolic models, which I'll call GEMS, and talk about how we can apply these to model soil communities and to predict their function. So genome scale models have been applied to soils before in a limited setting in lab conditions, and have been shown to capture the shifts in metabolism within individual soil aggregates, while they'll shift in their usage of carbon and nitrogen based off of availability of oxygen. But this has been shown only with a handful of species, I think about 4 species, and it's not clear whether this can predict the emergent properties in natural soils. And 2 of the obstacles to that I'll talk about today are that soil has a complex spatial structure, but we don't want to have to model every single soil aggregate on the planet. And most soil microbes are understudied and uncultured. So we don't have those high quality genome scale models that Dylan talked about, all the work that goes into experimentally validating the growth rate predictions for each of those models. So for our study system, we're comparing soils collected by the National Ecological Observatory Network, which has 18 sites that have data pairing microbial communities and their nitrogen fluxes. So we're looking at nitrogen fluxes as the emergent property of the community, and also the foundation of terrestrial ecosystems. So to talk about the first obstacle, the spatial structure, we're comparing a spatiotemporal approach with a non-spatiotemporal approach. So this one's called MICOM, or microbial communities, and it assumes that there's a trade-off between individual and community growth, but it's more of a static, steady state snapshot of a microbial ecosystem, which is very different from comets, which allows for a lot of variation in space. It allows us to set the diffusion parameters within a simulation to match how we think gas and nutrients are diffusing in soil based off of the soil texture, the nutrient availability, and the moisture. And then with both of these communities, with both of the approaches, we're modeling a community of 24 gems. That's 24 taxa, which is a mix of curated and automatically generated models. So that's what we use if we don't have access to the growth rate data to validate them. And I'll talk a little bit about the implications of that later. And then the outputs that we're comparing to, for comets, we have the actual predictions of 2 nitrogen fluxes, so nitrification and ammonification. And then for MyCom, we have these metabolic exchange scores, which represent the ratio of consumers to producers of specific metabolites like nitrate and ammonium, and we're using that as a proxy for the transformation rates of each of these nitrogen species. So for what we're seeing is that the simulations in comets are able to predict certain processes like nitrification pretty well, but aren't doing so well with the other flux we looked at, ammonification. But the key thing here was that it was still better than the non-spatiotemporal simulations. So including those variable diffusivity rates, also comets allows for localized interactions between taxa, which vary over space. And we think that modeling that spatial aspect of soil communities is going to be really essential to capturing their community properties. We also looked at how comets is able to capture habitat preferences. So these here are just plots showing how certain microbes prefer either the O or organic horizon or the mineral horizon of soils, which is deeper. And the comet simulations were able to predict what layer of soil the microbes prefer to live in based off of those environmental conditions. Next, we looked at how the curation of gems affected the community models. So we compared the manually curated models that went through all that experimental validation with the ones produced by software using template models. And we found differences in the metabolite niche similarity between microbes, where the red blobs here are showing microbes that had manually curated models, and they have different consumption of metabolites than the models that were automatically generated, which suggests that if we're only including one source, we're probably not getting the full metabolic diversity of communities, or that there's biases in the way these models are constructed by the automatically generation by the software. Now we'll look at how that translates to the actual community predictions. So for the flux in the top panel, ammonification, we saw that including the automatically generated models increased the error of simulations. But for nitrification, there was almost no difference between simulations that had either high quality models or had automatically generated models mixed into the community. And we think this has to do with how widespread the processes that the microbes in the community are carrying out. So nitrification was tied to just a couple of species that had really strong links to the nitrification cycle, to the transformation process. And those species had manually curated high-quality models, but ammonification didn't have just a couple of species that were the best predictors of those fluxes. It was a much more widespread process. So adding in the automatically generated models that were participating in those processes seemed to increase the error overall. So we think that this method also has a lot of potential for improving our understanding of how community composition links to function. And we want to make it more accessible, because right now you have to go through a lot of coding to get the models in a way where they can all talk to each other. So we've been working on creating a database of soil microbes that can be used in simulations. And this would let us assemble synthetic communities from soil taxa that have gems. And there are 26 manually curated gems available for soil taxa, and we have 67 taxa overall represented right now. And the example usage we're thinking is that if you wanted to look into microbes that were associated with certain environmental conditions, which could be conditions similar to Mars, or in this example, associated with like a pH gradient, you'd be able to filter by that sort of condition, and then see whether a gem is available for your genus of interest, or how closely it's matched to the species you're interested in, or whether there's a need for the community to create a model to represent that function. Now, to do that, we had to create our own database because no existing database has genomes for soil taxa that span bacteria, fungi, and archaea. So ours is going to be the most comprehensive database of soil genomes. And then once we have the database of organisms, we have to match it to the actual abundances of the microbes in the ecosystem, and that's how we determine which microbes are linked to those environmental properties. So we're also using the NEON infrastructure for this, which has about 1,500 metagenomes, which is the microbial DNA that's matched to plant communities, soil chemistry, and climate. And that's how we end up with these gradients where microbes are matched to different environmental conditions. And the hope is that we'll be able to integrate this into the COMET Smart interface that Ilya talked about and that we'll also talk about soon. A quick thank you to all my collaborators at BU and elsewhere. If you want to hear more about microbial forecasting methods for the soil, I'll be talking more about this on Monday, June 3rd, for my PhD defense. And I think there's time for questions.
So for a person that does space science, so I don't know much about microbes, so microbes don't just grow on everything and everywhere. That's the sense of it all, right?
Yeah, so early theories of microbial ecology suggested that, that most microbes could grow everywhere and that just the environment would select for certain microbes in certain places, but that the microbes had the potential to grow everywhere. But there's just a lot of variation in preferences for which microbes can colonize a space that only might have certain carbon sources that are harder to degrade, whereas other microbes will be more adapted to simple carbon sources and might be the later ones to join a community. So similar to how plant ecosystems have successional dynamics, where the ones that colonize initially are different from the ones that can dominate the community later on, it's similar with microbes, where the communities change over time.
I'm curious, do you envisage this being doable also for fungal genome and databases?
Yeah, so for these simulations, I was using the database that only included the one at the bottom that didn't have fungi, but the next round of simulations will be including all 67 of the taxa. And then some of the fungi, just there's only a handful that were abundant in most soils, but there were a couple of genera like Moderator that seem to be really participating in both the ammonification process and nitrification, and I think those will change the predictions as well. Okay, thank you.
So part of this project was our collaboration with the Software Application and Innovation Lab here at the Hariri, and we have the director that will also take you to the booth to show you what marvels they have been doing for us. Will.
Good afternoon, everyone. Thank you for Marianna and the folks who are involved in this project and all who made this event possible for inviting us out to talk today. A slight departure from the content topic that you all have been getting today. I'm just here to present one of the core facilities that exists within the Hariri Institute for Computing, and that is the lab that I'm the director of, which is SAIL, or Software and Application and Innovation Lab. SAIL is a software service center here at BU. We have a professional staff of software developers, staff of around 14, and our main job is to develop software products mainly around full stack web and mobile applications for BU researchers here and beyond, but mostly our audience is here at BU. And some of our core program areas of focus, which our projects are under, are listed here. We have ed tech. I mean, you can read at your leisure, but from ed tech to data science all the way to natural sciences, those are the typical realm of projects that we have, programs we have projects within. And it allows us to work with this sort of array of projects here and programs here gives you an indication that we work with a lot of faculty at the university, range from journalism to engineering to public health to biology, you name it. And our core service offerings with not just in the software development field are listed here as well. So there's the project and product management guidance, proposal and grant writing guidance that's largely baked into my role here, staff augmentation for roles that require data science or software engineering components. We are sort of a context holder for software development for researchers who have labs here at BU. So you think about graduate students, undergraduate students who develop software for research. Students' nature are pretty transient, so they come and go in different cycles. And so how do you maintain a tool or maintain the context of that tool or that software over time? And SAIL exists to be part of that element as well. And in the software best practice consulting, we do some of that as well. Initial phases of projects where there is a need to maybe do a heavy prototyping with graduate student software development or undergrad student software development in the forefront, and then later down the line, push a product to clinical or other research trials that have a larger audience tied to it. And in the initial phases of that, we often get in on the consulting side just to make sure that when software is transitioned from the lab to us for more production quality, there's a seamless flow of work and understanding of the software basis there. It's a lot of information, I know. Here's more of a lot of information. So this is just a list of example SAIL technologies that we've developed over the course of the last few years and just in general, ranging from our own independent microservices to mobile applications. I mean, the majority of our mobile applications are on the digital health intervention side, multimodal data repositories for ingesting educational research, hardware and software integration for lab automation for synthetic biology, and one that we'll see today in the back of the room here where we have a demo of the Common Smart Interface, sort of our computation-based web application. So a web app that you can access, anyone can access, that has an underlying process running in the background or back end to inform or to give users exposure to something that they would normally have to run or have a larger process around running and instantiating on their own. Data visualization is another. We've done desktop apps for, I thought I saw someone here. And we also recently have been, as you are pretty sure aware, with the boom of the recent trend of leveraging AI and large language models, are creating a sort of a large language model as a service where we allow our members of SAIL, staff of SAIL, to stand up the infrastructure to support using large language models for many aspects of research. That's pretty much it. Just a quick high-level overview. I'll be back at the back as well for questions if you have them. But essentially, we'll have some folks representing from the Segre lab here, and as well as my lab at BU, answering your questions and showing you aspects of the demo, the product we developed. Are we entertaining questions, or we go straight? Okay. If there are questions, let me know. Happy to entertain them. But otherwise, we look forward to seeing you all during the break.
So anything that we need that website for, that's you're the guys to call, yes? Or if there is, I don't know, data that we need sorting or.
So I would think the first way I would think about it is that if there is a software need that extends beyond your lab that you want others to access and interact with that supports research, that would be the primary element of focus that you would come to us looking for collaboration with. And we do general website development as well, but we collaborate a lot with other BU constituents for other elements. So typically an intake of projected work process, we look at the scope of work and what you're trying to accomplish. Oftentimes we'll take on the work directly. Oftentimes we'll funnel you to other institutes at BU, whether it be ISNT or Marcom or whomever. But usually the underlying focus is software that supports research in some kind of way. It could be directly through an application. It could be indirectly, even institute and center websites for research. We develop those in a typical fashion, but I think, yeah, the research element is the key focus there.
Thank you. And everyone, we are early, surprisingly, so we can start the break, and you can follow Will to see what they developed for us. They have a web designer in a way, because you designed the entire website, right?
Right. So it's a web app. We also design the user interface as well. We have design staff as well within SAIL we can leverage. Typically in research, you see there's a balance of function over fashion, with budgets being budgets for research. So you usually have folks prioritizing functionality first and then coming back to the look and feel. But this is a project example. We invested slightly more than average on the research front to the look and feel, as well as doing the functional requirements also.
Thank you. And everyone, please enjoy your break and follow Will.
Thank you.